not as good a turnout today. Here's the roll coming around, 8:30 front, 9:30 back. Yeah, this is probably the lowest we've had all semester. People just don't care anymore. Kind of not the worst session to not have very many people, though. It's probably going to be a shorter one. Today's material, not that it's not important or not difficult, but it's mostly memorization, vocab and type things. So I would just encourage you to memorize today's stuff. There's not like super difficult concepts. Here's the memes. This one always drives me nuts. I don't know why they couldn't spell checked that before they took the picture and posted it on the internet. This one always kills me. Anyway, we're going to do a quick review of some of the Chapter 17 stuff, and then we will get into today's Chapter 18. And obviously, any questions you guys have for anything related to Exam 4, which opens the week after Thanksgiving break. So that one always sucks. Don't let Thanksgiving break lull you into the sense of security, because you get back and the test opens the next day. So don't go forgetting about that. Which also means my super session will probably have to be the Monday before Thanksgiving break. So you may feel like that's like super way early before the test, but that's the only way we can do it. Since I would do mine Monday, Hillary would do hers Tuesday, and Chase would do his the Monday before the test. So those will be your only 3 super session opportunities. So again, don't let the Thanksgiving break make you feel like you can slow down. Our biology fact for the day: your nose can remember 50,000 different scents. And your sense of smell is actually the only part of your brain with like an entire region designated specifically for smell, right? You have a spot right here in the front of your brain called the olfactory bulb, and it only does smell, right? Like a lot of parts of your brain do like vision and speech, or like coordination and breathing, and like a few things, but that part just does smell. And it just goes straight from your nose to the olfactory bulb. So that's why smell is actually one of those really fast senses. Have you ever noticed you're like walking down the street and you're like, that smells exactly like October 12th, 2012. And there was like chocolate chip cookies in my grandma's basement and we were playing Super Smash Bros. Like, you know exactly what you were doing, right? It's just because you have that specific area of your brain designated just to that. And it can keep track of 50,000 very distinct smells, right? Like you will remember where you were and what you were doing when you smelled that thing, right? I don't know why. There's no like biological advantage to that. Like, it doesn't really make you a better, I guess maybe we would have been better hunters. I don't know. Our study tip for the day is to do a little physical activity before you study, maybe some Tai Chi. It's just because if you've been sitting in class all day and then you go home and you sit down and you just sit all day, it's not very good for you. You should be exercising. But also, if you're going to have like a 2-hour study session, if you just do like 10 push-ups or just run around the block or just do some sit-ups or run up and down the stairs a couple of times, whatever, it's a good chance to kind of get in the exercise before you just sit there for 2 hours, and sitting there doesn't feel as long, right? And your blood flows better, your brain runs on blood, so you're going to learn better, right? All of these things are connected, okay? Plus, you activate more areas of your brain when you move around, so move around while you study if that's feasible for you. Okay, you probably saw in the announcement this morning, I want to make a practice final, but I also have like 6 finals of my own. And so I honestly just don't have the time nor the ability to make an entire practice final. But as a group, we do. So I'm just asking that you make up even one test type question that you think you could see something like that being on the final. and email them in, parkerweber33 Gmail. I've had only one person so far do it, but there were some pretty good ones. And I'm just going to compile them, and obviously I'll make up my own as well. And if I get questions that I go, that's really not something that we would see on the final, don't feel offended that I didn't put your question in there. I don't want to, focus on stuff that's probably not going to be in the final. Also, if your question has false information, I'm not going to put it in there. So try to be, try to be accurate with what you're doing. Also, put the answer to your question in there, because I'll be honest with you, sometimes I'm actually not sure. So having the answer that you think is correct to your own question also helps. And that way we'll have a very good practice final that everybody can use, right? I know I love having practice tests. If you don't, then don't participate. Oh well. But I love having practice tests. I take as many as I can, and so if we all can kind of make one together, it will be very good. That's my standard email, so it's on the Canvas page. And it doesn't have to be today, because obviously we still have like a month of material left, but at some point email in a question or two, and yeah. Does that sound like a good idea to you guys? Is that something that will help? Got like 2 people to nod. If I make a practice final, will you take it? Okay. That means you have to help, because I'm not going to make the whole thing by myself. I'll just be frank with you. All right. Questions before we review some Chapter 17 stuff? I promise we will get to Chapter 18 stuff. Questions like from Mastering Bio. Questions on clicker questions.
50 questions on Mastering Bio, and you didn't cover like half of it.
Yeah. That's just standard. That's pretty standard. Google it. That's what I always did, and I never felt bad about it. I think Dr. Mott himself has told you guys to do it, because the way that I visualize it, the way that I think about it is I could get it wrong and then learn why I got it wrong, or I could Google it, get it right, and learn the right answer, right? So either way, I'm going to learn what's right, but in one way I get the points, and in the other way I don't. So don't just get it wrong because you don't know it. Google it. If you can't find it on Google, I'm more than happy to help you out. I've throughout the semester, I've had lots of people email me screenshots. It helps if you email a screenshot of the question and the possible answers, because sometimes I have to remember back quite a ways to what it is, and if the possible choices are there, that helps. There have been a few where they are like, what's the answer? And I'm like, well, I think it's this. Is that an option? And they're like, no. And I'm like, well, what are the options? So send the whole question, all of it. Any questions from Mastering Bio that you haven't? No? All right, we're just going to start on material then. We may get out early. I know you don't want to, but. All right, so 3 types of base pair substitution mutations. First one being silent. Why do we call it a silent mutation?
Because it doesn't physically affect like the phenotype. It's basically, what is it, like a degenerate form of the codon?
Yeah. So we change the sequence, but it happens to, in that case, code for the same protein that would have been there anyway. So yeah, it was a mutation, but nothing actually changed in the product, right, in the actual phenotype. Okay, how does that happen? How can we change the order but still get the same protein?
Multiple codons for the same protein.
Yeah, we call it redundancy, right, or degeneracy. The multiple sequences can code for the same thing, right? I can't think of any off the top of my head, and I don't have the codon table, but just throwing one out there, this is probably not accurate, but like CCC and CCA and CCT may all code for like leucine, right? And so if we change CCC to CCA, we're still going to get leucine. So you're not actually going to see any change in the protein, therefore you're not going to see any change in the phenotype, right? So it's not going to like change your hair color. Okay? Questions on, yeah?
So with these,
Yeah, those are the next 2 slides, so we'll hit those for sure. But yeah, so there's nonsense and missense coming up as well. Silence, just if we get the same amino acid, so the same protein, same phenotype. Other questions? No? So missense being one of the other ones. Somebody explain to me how a missense mutation occurs. It's like 30 of you in this room. You can't all stare at your notebook and act like I'm not up here.
Just codes for a different amino acid, changes the protein.
Yeah. So we do the same thing, right? We substitute a different letter. So if I'm looking at this right, yeah, we had a C here, so we put in a T there. So instead of a G, we got an A, which means we got an A down here. And so AGC instead of GGC gave us serine instead of glycine. So now it's actually a different amino acid, which means we are going to get a different protein, right? So we are going to see a change in phenotype. I don't know what this specific one would be. I don't know if it would change your hair color or your eye color. Could be a whole lot worse. You could end up with, sickle cell anemia or something, but something is going to deviate from what it should have, right? Cancer, for instance. Fun fact, sometimes after chemotherapy, because it irradiates all of your cells and destroys all kinds of stuff, your hair color and density and type can change. So people with curly hair will go in, get chemotherapy, lose all their hair, and they'll grow back straight hair that's a different color. You can totally change your hair color. So I just thought that's kind of cool. You go get chemo and all of a sudden you have a fro. Questions on missense mutations? Pregnancy, actually, that happens sometimes too. That's more the hormones than the DNA. A lot of women will end up with a different texture to their hair after they give birth. Yeah, it's not going to do it to your dreads.
All right, answer it. Forget. No. So the missense doesn't always change the phenotype, right? Sometimes it can, sometimes it won't. Is that true?
I would say that more often than not it will change the phenotype because you are changing an entire amino acid, and each protein is pretty specific, right? This and then that and then this and then that and then this makes that protein. And then if you start, even just changing one amino acid, it's like changing one ingredient in a cake, right? If you switch eggs for, applesauce, you're not going to get the same cake, right? It could be close, may not be drastic. I think you can actually substitute applesauce for oil, I want to say. I think I've done that before. And you get pretty close, but it's a little bit different. But pretty much there's almost no way to have a full missense mutation and not get at least some change. But oftentimes it's not massive, right? It may just be change in hair color, right? It doesn't necessarily have to be life-altering. But it is a permanent change. Other questions? Are you astounded by my knowledge of baking? It's because we started making brownies and we were like, we don't have vegetable oil. So we Googled it and they were like, applesauce. And I was like, well, all right. That wasn't great, but we had brownies, so. They just were almost a little bit mushier, like applesauce. I should have seen it coming. All right. Nonsense mutations. Somebody else tell me how these happen. Let's go here.
Isn't when you change the base pair, it's going to have a premature stop?
Yes. Elaborate a little bit. You're right.
So when you change it because AU changes the codon first, I don't know how to go much more. I just know that it stops before it's supposed to.
So yeah, we have a T in our original DNA, right? How things should have been. We switch in an A instead, right? Which means that codes for a T right here, which means that codes for a U right there. We had coded for AAG, right? That codes for lysine. Instead, we're now coding UAG. That's a stop codon the protein synthesis gets shut down, right? In this case, it got shut down immediately, right? That methionine is the start codon, and then we got a stop codon right away. Doesn't necessarily have to happen right after the start, but sometime earlier than the stop we would have wanted, a change in the base pairs codes for a stop, and you cut your protein short. This can be pretty problematic, right? If you stop coding for the proteins that make the valves in your heart, this could be an issue, right? If you stop coding for the proteins that build your spinal cord, going to be a problem. If you stop synthesizing the proteins that give you eyebrows, I mean, you're going to look weird, but it's not the end of the world, right? So again, there's always a range to these. I'm not saying this one kills you and this one doesn't, and this, there's all kinds of ranges, and I don't know enough about these to say like this certain disease is this and that and this, and I don't know. But you just need to understand a nonsense mutation is a premature stop codon, right? But you have to understand it's because we changed a base pair, and something that used to code for an amino acid now codes for a stop, right? Does everybody follow me on that one? Does anybody have questions with nonsense mutations? So if you're having trouble remembering them, silent means you can't hear it, so it's kind of like you can't see it, right? It's a silent mutation, so it happens, but it's like a tree falling in a forest when no one's around. Does it make a noise? Technically, yes, but nobody's there, right? No mutation is observed. Missense, then I just go, if you're having a hard time with missense and nonsense, missense is like a miscommunication, right? A misunderstanding. You heard me a little bit wrong, and so I clarify, right? And it's usually not that big a deal. We just maybe got our words mixed up, right? Whereas nonsense makes no sense, and we're gonna get nothing out of this, right? So nonsense mutation just makes no sense. We get no protein. No, not the protein, not the full protein, right? Could be stopped immediately, could be stopped halfway. All right, everybody following me? We're having a slow day in here. All right, now there are two other kinds of point mutations. These are insertions and deletions. So a substitution, we didn't lengthen or shorten our DNA at all, right? We took one out and put one in. So it wasn't shortening anything or lengthening anything. It was just a switch, right? A to T, C to G, G to A, whatever. Any kind of switch where we put in one for the other, substitution, right? Base pair substitution. Insertion or deletion, we actually physically put an extra nucleotide into the sequence, making it a little bit longer, right? Or we forcibly remove an entire nucleotide, and it can, be multiple along your entire genome, and it shortens just one nucleotide. But again, I showed you the example of, and Dr. Pogorski showed the example of the cat ate the rat, right? If you take out one letter and move everything to the left one, doesn't make sense anymore, right? It's not a sentence because it shifts the entire thing. I used the example of War and Peace. If you just took out the T in the first word of the book and shifted every word to the left but still left the spaces, the entire book would be complete nonsense, right? Doesn't matter how long or how short. It could be a Dr. Seuss, Green Eggs and Ham book, right? It's going to completely destroy everything. It just ruins everything. I don't know how else to say it. So here's how an insertion works. We used to have TACTT, okay, and ongoing. Now we have TACATT. So it used to read AUG, AAG. Now it reads AUG, UAA. Stop codon. That doesn't necessarily have to be what happens. It could have just changed the protein. It could have been a stop codon. It could have been a stop codon later on, right? A lot of ways this can happen. But what makes this different from everything else is the act of adding an extra, inserting a nucleotide into the sequence, okay, and shifting everything to the right. It's called frame shift. Compare that to a deletion. We take out that A, so it used to be AAA. Now it's just AAC. So now our code, instead of UUU, is UUG. So instead of, I think that's phenine, I don't know what that one is, and leucine. Okay, so we've actually changed the protein. How is this different from a missense mutation?
There's no frame shift.
There is frame shift. In a missense, there's not.
Yeah.
Is that what you're going for? Okay. That's part of it. What else?
The whole chain of amino acids changed.
Yeah. So notice that, this phenine or whatever it is became leucine, but now this glycine also, it's like kind of letting me draw. That's weird. This glycine became whatever that is. I don't know the names for these very well. Whereas in a missense, we just changed that one amino acid. The rest are going to be fine, right? We just changed one, kind of stuck in the middle there. This changes every single other amino acid all the way down the rest of the protein. And so you can get something super crazy different, right? This is how those flies got legs instead of antenna, right? It completely changed the protein. So instead of coding for antenna out of their face, I guess, it became legs, right? You basically just destroy the previous protein and you have something totally different, okay? Questions up to there? Excellent. So now chapter 18 stuff. I have like 3 slides on chapter 18 stuff because so far we've kind of just talked about vocab, right? So I've gone ahead and bold, italicized, and underlined the vocab words. The clicker questions that we saw in class today basically boiled down to understanding the vocab, right? It was something along the lines of liver cells and brain cells, or whatever cells, have the same control elements or regulatory sequences. I can't remember which word he used, which phrase he used. And so how are they not the same thing? If they're all if they have the same control elements, why are liver cells not brain cells, and why are brain cells not liver cells? Huh?
Is it through gene expression?
Through gene expression, yeah. What facilitates that gene expression? Activators, right? So your control elements are, I don't know why it's like, can you shake an electronic pen to get the, apparently not. The control elements are just DNA, right? It's just a fancy way of saying DNA, right? It's a specific kind of DNA that is coding for whatever you're looking for in that specific area. But it's just a coding part of your DNA, right? Is my DNA in my brain different than the DNA in my liver? No. It's all the same DNA, right? My toenails have the same DNA as my spinal cord, as my heart, as everything, right? All your DNA is the same everywhere. It's the activators present in that area. So in my brain, I have specific activators that bind to the brain cell, to kind of oversimplify it, control elements, okay? And it turns on that set of DNA and says, become brain cells. But in my liver, different activators attach to different control elements or regulatory sequences and activate those and say, become liver cells, right? So you guys see how we can have the same DNA everywhere, but we get different cells? Two of you acknowledged me, so I'm going to assume that the other 28 of you also know what I'm saying. So, here's a visual of how that works, right? So on the left of this picture here, we have liver cell nucleus. The activators up there happen to bind to those control elements, and it gets into a position where it can interact with transcription factors. Transcription factors start the transcription of your DNA, which eventually leads to proteins, right? And in that case, you get liver cell proteins, and you build a liver, or you renew a liver, right? Lens, like in your eye, is further down on the DNA. You don't have the same activators. You have different activators. And those bind to that specific portion of the DNA that has the right control elements, and it connects to a transcription factor. You start transcription, you build a protein, and you get lens proteins, and it builds a lens in your eye. Okay? It's all the same DNA all the way through. Okay? So the key vocab word is differential gene expression, right? It's basically just saying this is how we express for the gene we want in this area versus that area. This is why we get toenails on our toes and hair on our head and blood cells in our bloodstream and bone marrow in our bones. It's all made up of the same DNA, but the activators decide what it becomes. Okay? You guys following me on that? Did the clicker questions from class make a little bit more sense now that you understand? Because it was like basically like a 50-50 split, true and false. Hopefully all of you will now know which way to go.
I don't know if we'll need to know this for this class, but where do the activators come from?
I don't know. So I'm going to eventually guess that you don't need to either. Yeah, I have no idea. That's a good question. Some higher power puts them where they're supposed to be. Your God of your choice. All right. Alternative splicing. Somebody give me a rundown of what the heck that is.
In this one, you cut the same segment of mRNA or free mRNA into different segments depending on what gene you want to express, and then just recombine them. So you can pretty much just mix and match which pieces you want to create whatever product you want,
Yeah.
or what protein you want.
Exactly. So we have our original DNA, and it's got, in this case, 5 exons, right? We can splice this any way we want. We can splice it so that it's 1, 2, 3, and 5. We can splice it so it's 1, 2, 4, and 5. We could splice it so it's 1, 3, 4, and 5. We can splice it so it's just 1 and 2, 1 and 5, 1 and 4, 2 and 4, right? There's, I don't know super good math, but I think it's like 5 to the 5th power. I don't know how to do probability. There's a lot of options in how you could splice this, right? Especially considering that you could splice it, in a combo of 4 or 3 or 5 or 2 or just 1. And so what does that allow us to do? Because, yeah, it's all great and fine that we know that you can do that, but why would we do it anyway?
To express different genes.
To what?
To express different genes.
To express different genes, right? So if 1, 2, 3, 4, and 5 as a combination are going to give us some genetic sequence, right? So we have 1, 2, 3, and 5. Let's say that codes for like blue eyes. And then 1, 2, 4, and 5 codes for green eyes, right? It's obviously not how it works, but I'm just, throwing out examples, kind of off the top of my head, obviously. You splice to the left, you get blue eyes. You splice to the right, you get green eyes. Okay? You splice to the left, you get brown hair. You splice to the right, you get blonde hair. You splice to the left, you're a little bit taller. Splice to the right, you're a little bit shorter. There's all kind, and it doesn't even boil down to just that, right? Skin tone. There's a whole spectrum of skin tones. 1, 2, 3, 4 could be different than 2, 3, 4, 5. Then different from 1, 2, 3. Different from 2, 4, 5. There's so many different ways to do this that allow so many different genes to be expressed.
So does the spliceosome do it on purpose? Is it told you're gonna take 1, 3, and 5 and splice them? They just random?
I think it depends on the situation that you're in. Sometimes it can be random, and sometimes it can be deliberate. So if you're like, let's make a liver, then you splice it so that you get that right combo. And then obviously you introduce mutation. If something's wrong with your spliceosome, it's going to do whatever it wants. So it's not like a perfect prediction, but most of the time it's on purpose.
So this might be more broad, and I may have missed the point here, but yes, we all have the same DNA sequences, and obviously we have the different parts of ourselves through different gene expressions. What facilitates or helps like the DNA recognize that, hey, we need to be making different cells? Does that make sense?
So you're saying like how does my body know, let's do a liver here and a heart there and lungs here, and let's do brown hair, not blonde hair? Is that what you're saying?
Basically, yeah.
It's a very broad question. Part of it will actually be covered on Friday when we talk about further differentiation, so I won't get too ahead of myself. A lot of it is just, again, the activators that are present in that area. So down in your liver area, you have the activators to start the transcription of liver proteins, right? And in your brain, you have different activators to do brain proteins, right? I don't know how the body puts which ones where. That probably goes beyond the scope of this class. You're obviously welcome to Google it if you want to know. And then we'll cover the rest on Friday, but it's basically nearby cells say, hey, I'm a skin cell and you're right next to me. You should probably also be a skin cell. And it goes, yeah, I guess that makes sense. We don't want like a hole in our arm. And so a lot of it is just that interaction of neighboring cells. But we'll talk about that Friday. I hate to like not answer the whole question, but I don't want to steal Pogorski's thunder, mostly because he's better at this than I am. Other questions? That's it. Only 35 minutes. Yeah, so if you guys don't really have anything else, you may leave. You're free to leave whenever you want. If you do have questions, you can come up and ask me. You can email me. Try and email me some test type questions that you would want to see on a practice final. And I'll get working on that and have it up around the time that it's time to start studying for the final.